Launen from Keene State College, and this is a little short video that I'm making for you about the topic of meiosis. Meiosis is a cellular process that happens in eukaryotic cells, and it only happens in certain cells, which are referred to generically as myocytes. The purpose of meiosis is to begin typically with a diploid cell and have the ploidy, in other words, go from diploid to haploid, and also introduce genetic diversity, all for the purpose of making gametes that are different or somewhat different from the parental organism. As a side note, you should realize that the process of meiosis evolved from the process of mitosis. So the similarity is not accidental, but they do accomplish different things in the life cycles of eukaryotic organisms. Meiosis occurs in two stages, meiosis 1 and meiosis 2. Within each of those two stages are the substages prophase, metaphase, anaphase, and telophase. Shown in this particular image, prophase is not demonstrated, but it was occurring a moment before metaphase 1. So before any of this starts, what happens is the cell, the myocyte, undergoes S phase during interphase, and in S phase, the cell copies the chromosome or chromosomes. Those chromosomes remain attached to one another in structures that are referred to as sister chromatids, and that is what is shown in these X-like structures here. Each of these is a sister chromatid. So there are two sister chromatids shown on the metaphase 1 plate here. Blue and red are denoting the homologs inherited paternally and maternally, or vice versa, you pick. And so we have the two homologs for this long pair of chromosomes, the two homologs for these short pair of chromosomes, and they're in their sister chromatid state. So before all this happened, we had S phase forming the sister chromatids, then prophase 1 happened, and during that, the spindle fibers attached at the centromere regions to the sister chromatids, organizing and pulling them into metaphase 1. And in metaphase 1, what happens is these align on the metaphase plate, and then in anaphase 1, the homologs, still in their sister chromatid state, are pulled apart due to the shortening or condensation of the spindle fibers. As they actually make it to the poles of the cells, and a nuclear membrane begins to reform, shown here and here, we call that telophase 1, and then we go through the first cell division. So one cell has become two cells in meiosis 1. The DNA content is already halved, the ploidy is halved, so that has been addressed. In meiosis 2, so here are the two cells from meiosis 1, and they're coming in, and they would have also gone through prophase 2 and prophase 2, where they start to get organized and the spindle fibers begin pulling, they attach at the centromere region and they pull the sister chromatids along the metaphase 2 plate. And now you'll notice that the homologs in their sister chromatid state are organized end for end, as is seen in mitosis. This looks a lot more similar to mitosis. So they're pulled to the metaphase plate, that's metaphase 2, and then the sister chromatids at this stage are actually torn apart in anaphase 2, and each sister chromatid will make its way to a pole of the cell due to the action of the spindle fibers. Nuclear membranes start to form, and we call that then telophase. The chromosomal content is separated to the poles of the cell, and then a round of complete cell division happens. And so where one diploid cell went into meiosis, the result is 4 haploid cells, and each of these haploid cells is a pre-gamete. It will differentiate and become a final gamete. And it should contain one chromosome from each pair of homologous chromosomes. And we'll talk a little bit about the variety that you see in here next. So some really important things happen in meiosis with regards to the introduction of genetic diversity. During prophase 1 of meiosis, the sister chromatids that represent each homolog from a pair of chromosomes find one another, pair, and tightly adhere to one another in a process called synapse. And that's mediated by a lot of protein activity. And they do that so that they can trade equivalent or reciprocal sections of DNA in a process called recombination. So here we're seeing this. Here are the two homologs in their sister chromatid state. This isn't showing the tight synapsing. It's just focusing on the fact that we're getting crossing over in this region. And now we've actually shuffled up some of the chromosomes that would go through here or through here. And that is genetically different from what the parent to this organism would have had in its possible gametes. So this represents the introduction of genetic diversity through crossing over, happens during prophase 1 of meiosis. And it introduces new combinations of alleles along the chromosomes. Another really important aspect of meiosis 1 that allows for the introduction of genetic diversity in the organism going through meiosis relative to its parents is the fact that the homologs can line up on the metaphase plate in metaphase 1 in different possible configurations. So this is just focusing on a scenario where there are two pairs of chromosomes, long and short. Here are the homologs in their sister chromatid states. They are aligned on the metaphase 1 plate, and it's going up and down in this image. And if we say that red is the maternally inherited homolog, blue is the paternally inherited homolog, as this key is showing, then what we see is that you can have this possible configuration or this possible configuration. Each will occur at the same frequency. They are random. How long chromosomes assort themselves or place themselves on the metaphase plate does not dictate how the short chromosomes place themselves on the metaphase plate. They are independent actions, independent of one another. And because there are these two possibilities, therefore you get different possibilities in the final gametes with regards to the maternal and paternal chromosomes and how they can assort into the final gametes. So this is also really important, and it happens in metaphase 1. So what does meiosis have to do with the Punnett square, which is a tool that we use to model offspring and what they could look like, what the probabilities of different kinds of offspring are in eukaryotic genetics problems? Well, meiosis is the first step in forming gametes. And gametes are what go along the axes in a Punnett square. So when we're making a Punnett square and we're saying, here's this organism, two genes, heterozygous for both gene pairs. It's big R, little y, little y. And we ask the question, well, maybe what would happen if this organism mated with another organism of the identical genotype? Well, if you want to model that with a Punnett square, you have to ask, what gametes can this double heterozygote make? And the gametes that it can make will include, there will be 4 types, and they're shown here. And it will include all of the possible combinations of the alleles of R and Y together. So big R, big Y, little y, big R, little y, and little r, big Y. And those are the 4 things that would be placed on the axis of a Punnett square. Next, we'll be talking about aneuploid conditions and non-disjunction in